Alright, we are doing this video outside, so it's going to be a little low tech here. We've got a clipboard, but we're going to make it work. This video is on genetics and inheritance patterns. So we're starting off with a review from 7th grade. If you didn't learn this, then you can learn pretty much all of 7th grade genetics in like 2 minutes here. So there was a guy named Gregor Mendel, and he was actually a monk with a science background. So when he was in the monastery, he was actually doing experiments, and he experimented with pea plants. He had 7 purebred pea plants that showed different traits. The reason he chose these plants is because they have a lot of traits that are pretty obvious to experiment with, and they reproduce fairly quickly. So what he did was I'm going to give one example. We're going to do flower color. So he had a parent generation, which we're going to call the pea generation, that had either white flower or purple flower. So, okay, parent generation. So flowers were either white or purple. Remember that these are purebred, okay? So what he did was he crossed the white and purple flowers, and we get the F1 generation, which is just our first offspring. What he found was that all of the plants in this next generation, the F1 generation, were purple. Purple. And finally, he crossed the F1 generation to create F2. And what he found was that they all weren't the same anymore. He actually got 3 purple, 1 white. So he looked at this and he wondered, here we go, why was this happening? Why did we start with this white trait? It disappears, and it reappears again in the F2 generation. Now he saw this with all of the different traits he was testing, so plant height, pod color, wrinkled or non-wrinkled, the shell around the piece of pod. What else did he do? Height, I don't think I mentioned height yet, and stem, he looked at the stem of the plants. And all of them, no matter what, got something like this, where in the F1 generation, it was lost and then refound. So what he started to figure out was that it wasn't necessarily lost, it just wasn't being expressed. So during his time, these ideas were not accepted. The common belief at this time was that you get half from mom, half from dad. It's a perfect mix of the two, and that's that. And so he was fighting this idea with this as his evidence. Now, throughout his entire life, even until he died, these ideas were not accepted. This came much later. However, to this day, we know him, Gregor Mendel, as the father of modern genetics. Now, I'm going to go into some vocab of what he actually discovered from, actually I'm going to put this back up, from these experiments. So he found that the purple flower here would be dominant. I'm just going to use A because it's easy to see on the screen. So both of the alleles for this gene are purple. And here for white, they're both white. So when he crossed them, and this is where meiosis comes into play here, because each parent gives one allele. So this plant is only giving one allele, and this plant is only giving one allele. So all of these purple plants have one from mom, one from dad, and are big A, little a. Now when we have something that looks like this, big A, little a, this is called heterozygous. In a heterozygous trait, the dominant the dominant trait is what's going to show. So in this case, only the purple shows because the purple is dominant over the white. But we get something different down here. We start to see that white again, so that means that we cannot have a dominant trait or a dominant allele in this plant because it's showing white. We know that that only happens with two little a's. Whenever we have the two little a's and the two big a's, this is homozygous, so they're both the same, okay? So this would be homozygous recessive, this is homozygous dominant, this is heterozygous. So, and I put that here for you guys. We have dominant, recessive. I'll go over that in a second. So what he did was he crossed big A, little a, and with big A, little a to see what we got. So this would be an example of how we would cross the F1 generation with itself, okay? So we have a Punnett square here. This is a basic Punnett square. This should be reviewed for you all, but if not, it's okay. How we do this, we traditionally put the alleles from mom on the top, the alleles from dad on the bottom. Remember, only one of these is actually, well, one from here and one from here is actually going to be passed along. So what we're doing by creating this Punnett square is we're seeing all the different possibilities of what the outcome could be for the offspring. So we're going to start by just bringing, I'm going to show this really basic, this top one down all the way. So bring big A here to here, and big A here, bring it down. Okay, now I'm going to do that with little a, same thing, I'm just bringing it down. Little a, little a. We usually put the lowercase second, so that's why I kind of put it at the end of the box there. Now we go across, okay? So big A goes here, big A over here. Little a here, little a here. So what we just did was we saw how any of these alleles could possibly cross with any of these alleles. So these are our 4 possible outcomes in the next generation. So some vocab here, we have, once again, dominant, recessive. This would be an example of heterozygous pairing. This would be an example of homozygous pairing. They're the same. So I'm going to go ahead and show you guys now how we determine genotype and phenotype from this and show how that relates back. So our genotype is basically sharing all the possible outcomes based off of their alleles. One of these would be an allele. This would be the gene. So our genotype, we have big A, little a, and little a, little a. So we're looking at all of these possible genotypes. We have 3 because these 2 are repeating. So our first one we have is one that has big A, big A. We're just creating a ratio here, that's all. Then we have 2 that are big A, little a. And then we have 1, little a, little a. So remember our genotype, remember that's the genes, how it actually is, like what it is made up of. So our genotype is 1 big A to 2 little big A, little a to 1 little a, little a. Now this is genotype. This is different than phenotype. Phenotype is going to be how it actually looks. So this is going to be that purple or white, not just what it's made up of, but what it's showing. So remember that when we have something that's heterozygous, so it's showing both big A, little a, what's actually going to be displayed is the dominant trait. So in this case, if we're using purple and white flowers again, we have 1, 2, 3 that are going to be purple because A is purple, oops, little a is white. Okay, so our phenotype, 3 purple to 1, that will be white. Okay. Once again, this is just our basic Punnett square. Genotype are actually writing out the different alleles, and then phenotype is the traits, what's actually displayed. So what we see now, if we go back to this, is that in his F2 generation, we had 3 purple and 1 white. Well, if you look here, we have 3 purple, 1 white. So what that means is that big A, little a was self-pollinated and crossed to give us our 3 purple and 1 white, and that is why that white flower kept popping back up in the F2 generation, or why the short pea plant kept popping up in the F2 generation, is because those alleles that code for it were not lost, they just were not being actually displayed. So you can see that all along they were actually there, just being hidden in a heterozygous pairing. Now, something else I wanted to discuss real quick are a couple of these other words here that I have. One is incomplete dominance and the other is codominance. So while we are on the theme of flower color here, we actually see this a lot in plants where we have something that's called incomplete dominance. So, for example, we could take a red flower and cross it with a white flower, and the phenotype for the offspring are pink. Well, that's because neither the red or the white take over. Neither one is going to be dominant over the other one. So instead of one being dominant, they actually share it. So those traits are going to blend. So that's going to be incomplete dominance. So your result would be the pink flower. However, in codominance, both are dominant and they're both shown. So if we had our red and white flower again, instead of having a pink flowered offspring, you would actually have both red and white showing on that flower. So you could have like lines going through it or something like that. That's going to be our codominance. Together, they are dominant. Incomplete dominance, neither one is fully dominant, so they just blend. Let's see what other words do I have on here. Polygenic inheritance is going to be, well, remember we have poly is just going to be multiple and genes here, so we have several genes that are going to control one trait. So while this is like nice and easy and it's like, oh, purple flower, white flower, it's not always going to be like this. Sometimes there are several genes that control a trait. So an example of that would be skin color. It's not always going to be perfect, nice and pretty like this. Okay, here we go. A couple of the laws that you're going to need to know for this, now this isn't review anymore, we have the law of segregation and the law of independent assortment, that these were two of the laws that Mendel came up with while he was doing these experiments. Now remember, when we have meiosis, we're getting our 4 daughter haploid cells. So instead of having big A, little a, you get either big A or little a to pass along. So that way when the egg is fertilized and becomes a zygote, you have now a complete set of chromosomes, which means you're going to have 2 genes per trait. So what we'll see here is the law of segregation shows that the parents' alleles are going to actually be split. So big A comes to these 2 and little a, let's say these are eggs, big A in these 2 eggs, little a in these 2 eggs. So if a different egg was fertilized, well, you would not have the same traits necessarily that you have now. The other law that he came up with was the law of independent assortment. And what that means is that the alleles are going to separate basically different from one another. So they aren't related. So just because you have brown eyes does not mean that you have to have brown hair. You can have brown eyes and blonde hair, or you can have blue eyes and blonde hair or brown hair. So basically it's showing that they are not connected necessarily. You can have a variety of these traits. They don't get passed along together as a group. They independently are sorted out. So when we are tracking one gene at a time, we are looking at a monohybrid cross. That's just your traditional Punnett square, what I just showed you. But we can also look at two genes at once in a dihybrid cross. So that's what I'm going to show you guys here. So, rubber pen. Sorry, it's super messy. What I did was I crossed two heterozygous genes. Okay, so I have one gene here, which is big A, little a, my second gene here, which is big B, little b. Each gene made up of two alleles, all right? So one allele is big A, little, and the other allele is little a. So in order to fill out all of this top and the side, you're just going to find every combination that you can here and put it across the top. So I did big A, big B, big A, little b, and then you do little a, big B, little a, little b. All of those combinations, those four, are written right here. Now I'm crossing it with the same exact thing on the left-hand side, so it's just the same. When we go through this process, though, it's the same as doing a monohybrid cross or a normal Punnett square. You're just going to put the two letters that are the same next to each other. So when you bring it down, just leave a little bit of space. So I brought down big A, here we go, big A, brought it down, big B, brought it down, and then same with the cross, big A here, big B here. You can follow that all the way through. Note this one is over here because I messed up and it's kind of messy. But when we cross two heterozygous genes in both a monohybrid and a dihybrid cross, we're going to get certain ratios for phenotypes. You need to be aware of those because this is what you're going to get quite often. So when we have a dihybrid cross, two heterozygous traits crossed with each other, we are going to get a 9:3 to 3:1 phenotype ratio. So what I did is I circled all of the blue, or I boxed all of the ones that are blue here. What you'll see is that all of them that are boxed in blue are going to be showing the dominant trait, which makes sense because it's dominant. So this one, even though it's big A, big A, big B, big B, will show the dominant trait for both A and B. Here we have big A, big B, little b, but we're still showing the dominant trait. So all of the ones that are boxed in blue are 9. Then we have our ones that are going to show the dominant trait for A, but the recessive trait for B are going to be boxed in red here. That's where we get our 3, and then we have the opposite. So you have the ones that are going to show the recessive trait for A, 1, 2, and 3, and the dominant trait for B. And finally we have recessive all the way across. So you can see it's really rare that it's going to be completely recessive. To get this, it's pretty low ratio. So our most common that we're going to get is the one from the 9, which is showing a dominant traits for both genes. What we've covered up to this point has been like our perfect textbook genetics. However, it's not always going to be like that in living organisms. So we have a lot of things that will occur that increase variation and will actually change how you look, so it's not always going to be that perfect cross. One of the things that we have is something called crossing over. Crossing over is going to occur in prophase 1 of meiosis, obviously. Now, we have in prophase 1, our tetrads are going to start forming. This is kind of in the middle of prophase 1 and metaphase 1. Our tetrads are when 2 of the homologous chromosomes start to line up. And what's actually going to happen is a portion, oops, wrong side, this side, not here, of the chromosome part one of the genes can actually flip. They'll kind of come together and they'll flip. So you can actually have phenotypes that are not shown in parents start to show due to crossing over. So our result of crossing over would be that most of this is still blue, you still have the same, but right here where we had crossing over, we're going to get something different. And same thing here. Mostly red still, with a little bit of blue. So that's actually going to change the alleles that you have on that particular chromosome. So we'll still have big A, we'll have big B, but then here, here, because of that crossing over, we're actually going to have little b now, and that's going to change what is passed on to the gametes. Because remember then, once we have the second round of meiosis, then each of these are going to be split. So it actually totally alters the possible outcomes. So here we'd have little a, little b, big B, little b. So before, where we were going to get basically 1, 2, 3, 4 cells with one chromosome each, now we have big A, big B, big A, little b, big B, little a, little b. So I'm going to write that out just so you can see that. This was the options before. For our 4 gametes. This would be 1, 2, 3, 4. However, now because of crossing over and this change, because remember these are sister chromatids, so these are just getting split in half in the second round of meiosis. So now we have big A, big B. And you can see how this creates variation in our genetics.